Okay, so today we're going to finish up talking about acid-base balance. And then we're going to move into some basics of the GI system. So remind me, you've already looked at the GI system in Anatomy Lab, right? Okay, great. So the beginning of our discussion of the GI system will probably be a fair bit of review. Okay, so when we were last talking, we left off thinking about the fact that we're trying to maintain specific ratios of acids and bases. And when we think acids, we're thinking hydrogen ions, but we're also thinking CO2 because it can ultimately be converted into hydrogen ions. And we're thinking about bicarbonate. So we know that we want this 20:1 sort of ratio of bicarbonate to our acid, in this case on the slide, CO2. We're trying to maintain this ratio, and we know that either the kidneys or the lungs can kind of go wrong here potentially. So our disturbances, we can either classify as respiratory or metabolic, in which case we can mean kidneys, but we're being a little vague, so we can include things like hemorrhage, any other non-respiratory things. And then when we compensate, we're going to compensate either by changing our respiration rate, by changing our breathing, and therefore regulating how much CO2 is in our blood, or we're using our kidneys to regulate how much of our bicarbonate we're reabsorbing, therefore putting more buffer into the bloodstream, or synthesizing. We're synthesizing that from carbon dioxide and water. And we're also controlling kind of as part of that process, how much hydrogen is secreted into our urine ultimately. So we're changing how much acid we're keeping in our plasma and how much bicarbonate we're pulling into our plasma in order to kind of buffer out extra acidity. So when we're trying to assess what's going on with a pH disturbance, first we're going to check whether we have an acidosis sort of situation, which would mean a pH below 7.35, or an alkalosis, which would be above 7.45. And then we're going to figure out the cause of that. So our first cause, respiratory acidosis, so a cause of a low pH due to respiration, would basically be that we're keeping too much CO2, which is a biological acid, in our plasma, in our blood. And the reason we would be keeping too much CO2 in our plasma is we're not breathing fast enough. So we're not breathing off all of that carbon dioxide. So ultimately, keeping too much CO2 in your blood is going to increase the number of hydrogen ions in your blood. And the way we'd want to compensate for that is using a different system. So we'd use renal compensation to adjust for respiratory acidosis. So we're basically going to pair our two systems. Right? If our respiratory system is the problem, our renal system is going to be the solution. So we're going to have renal compensation and vice versa. So in the renal compensation methods, the way we can adjust for this increased acidity is to increase our hydrogen ion secretion. When we say secretion, right, we're moving it from our plasma, from our cells, into the filtrate, into the urine, and then we're ultimately excreting those hydrogen ions that have been secreted, so we're getting rid of them. We can increase our bicarbonate reabsorption or bicarbonate synthesis as well. If we're in respiratory alkalosis, so our pH is too basic, it's above 7.45. This means that we actually don't have enough acid in our plasma, right? So this probably means that we have a decreased partial pressure of CO2, right? So not as much acid, means our pH is more basic. So we have too few hydrogen ions ultimately. So cause for this could be hyperventilation. You're breathing off too much CO2 because you're breathing so quickly. And again, because this is a respiratory issue, that's why we called it respiratory alkalosis, we'd use renal compensation to adjust. So in our renal compensation, we decrease the number of hydrogen ions we're secreting. And if we're not secreting them, that means they're staying in our plasma. plasma, because we want more of those hydrogen ions available, they're going to decrease our bicarbonate reabsorption. So on the flip side, if we have metabolic issues, so metabolic acidosis or metabolic alkalosis, usually this may, well, we're not going to specify necessarily what the causes here are, since this is kind of a grab bag of anything that's not respiratory. Okay. So the cause of metabolic acidosis is an increase in hydrogen ions for any reason that's independent of our CO2 regulation, which is why we're saying it's independent of our respiratory system. Okay. So our first compensation method is going to be using the respiratory system because that's going fine. And then our second sort of line of defense here is if it's not a renal problem, if it's like you ate something or you're bleeding, if it's an issue that doesn't have to do with your kidneys, then you can also do renal compensation. If it's an issue with your kidneys, you're not going to involve renal compensation here because that's the part of the system that broke. So in the respiratory compensation for metabolic acidosis, you're going to start breathing faster. You're going to increase your ventilation, meaning you're breathing off more CO2. So you're going to see decreased CO2 levels in the blood. Potentially, you're going to do renal compensation. So that would work the same way as it did for respiratory acidosis. You'd secrete more hydrogen ions, urinating them out. You'd increase your bicarbonate reabsorption and also increase any synthesis of new bicarbonate. In metabolic alkalosis, we have the reverse. So we have acidity, decreased hydrogen, or sorry, we have decreased acidity, so decreased hydrogen ions. And this isn't an issue with our carbon dioxide. It's not an issue with the respiratory system. So we're first going to compensate using the respiratory system. So we're going to decrease our ventilation. We're going to decrease our respiratory rate, meaning we're increasing that CO2. We're keeping more of that CO2 in the blood. An acid, so it's bringing the pH down. And then if the problem is not our kidneys, if it's not a renal problem, then we're going to do renal compensation as well. So we're going to stop secreting so many hydrogen ions. We're going to decrease the hydrogen ion secretion. And we're going to decrease our bicarbonate reabsorption and synthesis of new bicarbonate. Okay. So once you get a hang of this chart, I honestly find this one of the most useful sort of summaries of what's going on in our acid-base disturbances and compensation. And it's kind of like a step-by-step flow chart of how you would tell what you have going on. So you could start out by measuring the arterial pH, right? So that's where we would measure it. You take a blood draw, you measure the pH of the blood, and then you're going to get a number. If you're anywhere between 7.35 and 7.45, you're fine. That's our normal range. That's our desired range. But if you're below that 7.35, you've got acidosis of some type. If you're above, you have alkalosis of some type. So we're going to spend some time on the acidosis side first here. Okay. So we know we have a low pH. So we know this is an acidic sample. Our blood is too acidic. Now we want to figure out why, right? We want to figure out, is this a problem with breathing, or is this some other type of problem? So there are two things we can measure here. I find it easiest to kind of follow this pathway with the CO2 first. So that's what we're going to do. So we know we have acidosis. We're trying to figure out, is this a respiratory problem? The easy thing to do is to just start by looking at that partial pressure of carbon dioxide. If it is a respiratory problem, if it is a problem with our breathing, we're going to see abnormal levels of CO2. And specifically, we're going to see CO2 is too high. And the reason it's going to be too high here, right, is that CO2 is a biological acid and we know that we have acidosis. So when we see that that CO2 is too high and our pH is too low, we know that's the cause, right? So we can kind of almost stop here. We diagnose that we have respiratory acidosis, that the reason our pH is too low, the reason we've created this disturbance to our acid-base balance is because we have too much carbon dioxide. And so, since that's a problem with the respiratory system, we're going to do renal compensation. And the way we do renal compensation for this is largely by bringing in more of our buffer for acidity, which is our bicarbonate ions. So we're going to see that we see more bicarbonate than usual, right? Because it's kind of balancing out that carbon dioxide. So we're going to see that our bicarbonate concentration is above 24. Okay. You'll see that number 24 is really what we're comparing to throughout this chart. So that's a good one to kind of keep in mind. All right. So on the flip side, let's say we know we're in acidosis, but we measure the CO2, and it's not above 40. It's probably even going to be below 40 in this situation. We'll see when we get to the bottom. Okay. So what's happening here, right, on this other side, is that the reason we are in acidosis doesn't have to do with the carbon dioxide, has to do with the fact that we don't have enough of our buffer, right? So when we measure the concentration of our buffer, our bicarbonate, we're going to see it's too low. So it's lower than this 24. And that's the reason we're in acidosis, right? Like we have the usual, essentially, levels of acid kind of peeking through, right? Basically, because we don't have enough buffer, we don't have enough of that chemical sponge to soak them all up. So this is an indicator that we're in metabolic acidosis, okay? So the first thing that's going to happen for sure is our respiratory compensation. So in order to control for this fact that we don't have enough of our bicarbonate, we don't have enough buffer, we're going to try and decrease the amount of acid we have in the system. So we're going to see that we're actually going to breathe faster, and we're going to drop our CO2 below its normal partial pressure of 40. And this is to kind of compensate for this decrease in buffer. So that's the sort of first way we're adjusting this situation. Questions about this side? So as you go through, I would practice spitting out kind of numbers at each other or at yourself for a pH, a bicarbonate concentration, and a partial pressure of carbon dioxide. Because using these three numbers, you should be able to sort of step through this chart, first on paper and then in your minds, to figure out what type of acidosis or alkalosis you have, and then what type of compensation is going on. So on the alkalosis side, right, we have the reverse. If we have not enough carbon dioxide, an acid, this means our alkalosis is due to the respiratory system. So we have respiratory alkalosis, which means we're compensating using our renal system. So we notice that because we have not enough acid, we're also going to decrease the amount of our buffer bicarbonate so that we are in balance, so that we're keeping that 20-to-1 ratio consistent. Or if our alkalosis is due to the fact that you swallowed a bunch of baking soda or something, so we have high amounts of bicarbonate, that would be metabolic alkalosis. We're going to adjust with our breathing. We're going to do respiratory compensation. So we're going to slow down our breathing to keep more carbon dioxide in our plasma. So we'll end up with higher levels of carbon dioxide to match those higher levels of bicarbonate. Again, keeping that 20-to-1 bicarbonate to carbon dioxide ratio kind of in check. So that's how you use this chart. And I recommend studying with this a little bit, and we'll come back and ask some questions about this when we review as well. It's supposed to be all of them. I'm having so many tech issues today. Slides. So now we're going to move into our GI system. So we're going to be thinking about the structure of our GI system. So it doesn't look weird. And we're going to be thinking about what's happening in the different parts of our GI system. And we're going to focus on basically the mechanisms that allow us to digest food. So that's kind of the big takeaway we ultimately want to get from this unit. We've covered the anatomy, at least the gross anatomy in lab. So now we want to understand what's happening in these different places. So we're going to start by going through the anatomy, but we're going to tack on what you've already done in lab. We're going to tack on thinking about the cells in these different regions and some of our tissue layers. Basically, our overview of why we need to understand these gastrointestinal system processes. But like you eat a burger, right? The nutrients you're eating are too large to enter into the bloodstream. Or say you're eating some broccoli, right? Like you can't take a head of broccoli and just move it into your plasma. So you're going to need to break that down both physically and chemically to small enough pieces that they're going to be able to move across your cells and into the bloodstream. So we have a lot of complicated things that are going on throughout our GI system that have to do both with like mechanical movement and mechanically breaking things down. So think like chewing, right? That's part of your digestive system, as well as breaking it down from a chemical perspective. We're going to think a lot about different enzymes that break down different types of molecules so that we're ultimately able to absorb nutrients. So we're going to be thinking both about movement, and usually when we're thinking about the gut, chewing does happen at the top of our GI system, but as we move through most of the GI system, we're going to be thinking about something called motility. And that's basically just the movement of like kind of squeezing things along, squeezing things through this series of tubes. We're going to be thinking about secretion. So secretion, when we think about the GI system, we're mainly thinking about creating enzymes and putting them into the lumen, so putting them into the contents of the tube in order to first digest our nutrients. So in order to break down your food into small enough pieces, and then ultimately absorb our different types of macromolecules. So brief review of what counts as part of our GI system. Right, so our GI system is a hollow tube, which means we're going to be thinking about the walls of the system, as well as the lumen, just like when we thought about the nephron. Right, we have a hollow tube, about 15 feet long, begins up at the top with your mouth, bounces apart. So we move through the mouth into the pharynx, which we sort of colloquially think of as the throat, I would say, and then into the esophagus, which is passing behind your trachea, behind, posterior to your lungs, entering into your stomach, And then from the stomach, we move into the small intestine. So our duodenum, our jejunum, and our ileum. And then into our colon, our ascending, transverse, descending, sigmoid, and then into the rectum and the anus. So we have a hollow tube that goes all the way through you from your mouth to your anus. So that's the first part of our GI system. That's the part we call our GI tract. But also important to this whole process are our glands. So we call them the accessory glands. And these are basically where our enzymes are coming from. So when we talk about chemically digesting our food, this is why we need those glands. We need to get those enzymes from somewhere. Sometimes we're going to have enzymes in the walls of the tube, but we're often going to have to secrete enzymes into different parts of our GI tract in order to break up the contents. So we're going to go through, basically we're going to go through the GI system in several different ways. We'll go through it regionally, and we'll talk about the different parts of the GI tract, different accessory glands, but then we're also going to go through it by macromolecule type. So we'll talk about, okay, so how do you digest a carbohydrate versus how do you digest a protein versus how do you digest a fat? I'll try to key it in as we go through on parts that you'll see later. First, we're going to look at just the sort of basic layers of our GI tract, so what we have in the walls. All right, so up until now, we've talked about a lot of tubes that have really thin walls where we just basically have an epithelial layer, an endothelial layer that we're interested in. But when we're thinking about the gut, we're going to have more that we need to do here. So we're going to see that we have a lining that's directly in contact. We're going to have interspersed layers of different types of muscle and connective tissue. And we're also going to have multiple plexuses of nerves kind of embedded in parts of these walls so that you can control the activity of the gut. So this list starts from inside to out. So we're starting with what's in contact with the lumen. We're going to get deep into sort of all the layers within these layers. Okay, but the basic thing we're looking at, the lumen is where your food is passing, right? So that's the inside. Then we're going to have our mucosa. So our mucosa here is in yellow. And this is the lining of the lumen. And as the word mucosa might tell you, one of the important things here is that we're going to have a mucous membrane. Then we have the submucosa, which is a connective tissue layer, but we're going to see that we have some glands in here. We have some other things going through, including one of our plexuses here. Then we have a layer of smooth muscle, muscularis externa, and then we have a layer of connective tissue kind of on the outside, rounding it all out, the serosa, which is basically what you would have been touching when you were in lab. So we're going to start with the mucosa, so that innermost layer, that mucous membrane. Okay. So what this is doing is separating the contents of our GI lumen, so separating our food, essentially, from the internal environment, by which we mean like from the rest of the abdomen, all the other stuff in there. So the first part of this is going to be the direct cell lining, right? So this mucous membrane here is made up of some specialized epithelial cells that we call enterocytes. So entero, I don't definitely know what that means. I assume it means like inside, probably site cells. Okay. So we're going to have absorptive cells, and their focus is going to be actively bringing the nutrients out of the lumen and into your body. we're going to have exocrine cells. These exocrine cells are going to be responsible for secreting stuff locally. So one of the things they're going to be secreting is actual mucus, for example, to line this pathway. So those are our exocrine cells. And then we're also going to see that there are hormones in our gut that help us control how active our gut is, how motile it is. Some of these are also related to things like hunger cues. So there are also going to be endocrine cells within this mucous membrane layer of the mucosa. There are other parts here within the mucosa. So we also have a connective layer. So that's here, basically, called the lamina propria. And then we're going to have a thin layer of smooth muscle called the muscularis mucosae. So what you don't want to do is confuse this muscularis mucosae with our outer layer of muscle, the muscularis externa. So we have two layers of muscle, one of which is part of this inner mucus layer. So if we're thinking about that lamina propria, it's made of connective tissue. Things we would see traveling within it are things like small blood vessels and lymphatic vessels. We're going to talk about the importance of lymphatic vessels to the absorption of fats in particular. We're also going to see some nerves in here. We're going to see some lymphoid tissue. So it is the case that when you eat food, right, you may very well be eating pathogens. You're definitely eating all sorts of bacteria. So it's important to be able to combat those pathogens you may be taking in as part of your digestive system. So that is the role of the lymphoid tissue here in the gastrointestinal tract. So this is contained within lymph nodules, these sort of like patches of lymphoid tissue called Peyer's patches. So those are still within this mucosa layer, basically. Then we have that thin layer of smooth muscle. The point of this more inner layer of smooth muscle, the muscularis mucosae, is to help you mix around the luminal contents. So we're going to want to churn our food, basically, so that it comes in contact with those cells in the mucous membrane, right? We want all of our food, preferably, to come into contact with our absorptive cells for sure, right? If our contents of our lumen, if our food doesn't touch the walls of the GI tract, then it's never going to touch those cells and we're never going to absorb it. We'll also see that there are some enzymes that are kind of embedded in that wall in the enterocytes. So in order to chemically digest food, again, we need to be able to mix that luminal content so it comes in contact with the walls. So this muscularis mucosae has a really important job. It does this by having both circular and longitudinal fibers so that it can sort of jiggle the food in both directions. And it's just doing this mixing kind of churning type of action. So that brings us to our next sort of big layer, the one in purple. So this is the thicker layer of connective tissue, our submucosa. And its main role is to allow your GI tract to basically change shape depending on what's in it, right? So this layer of connective tissue provides the GI tract with this property of distendability. So distendability meaning basically that it can expand, that it can stretch as you have more contents there. And elasticity, which we've talked about before, kind of the tendency of something to snap back, basically. So in our daily lives, we think of elasticity as that stretchy bit, but when we're thinking about actual physical properties, when we say elasticity, some of what we're talking about is that kind of rebound effect. So we want to be able to stretch out our GI tract, but we don't want it to just stay stretched out forever. We want it to shrink back down when there's not food. There are, again, blood vessels and some lymphatic vessels here in this layer of connective tissue, which if you think about it, kind of makes sense. If you have blood vessels in that innermost layer, they gotta go somewhere, right? They gotta get there somehow too. So they're also traveling through the submucosa. And we have a pretty important nervous plexus here. So we're going to have two nervous plexuses that are in communication with each other in the walls of the GI tract. So together they're going to make up something called the enteric nervous system. It's like kind of its own sort of branch of the nervous system that's in the walls of an organ, in this case specifically the GI tract. So this first one we could call the submucosal plexus, which is a little easier to remember because we're just naming it anatomically for where it is. But you'll often see still terms referring to the people who found them, or at least who wrote it down. So this is often referred to as Meissner's plexus. So that's what these first little yellow threads are indicating.
Part of the that like would have floating, or would that be more inside
I'm not sure what you mean.
like when you
Mhm.
you like get bloated, and so is that part of the extendability and elasticity?
Oh, yeah, that would have to do with the descendability. It would be distended. And I think bloating is often like collections of gas within the lumen. Yeah. Yep. So not in the submucosa specifically, but like within the contents of the tract. So that brings us to our bigger muscle layer. So far we've seen the mucosa, which is our innermost layer with that mucous membrane, the lamina propria, and that first little thin layer of smooth muscle, the muscularis mucosae. Then we had a big connective tissue layer, the submucosa. So now we're on our larger, heftier muscular layer, the muscularis interna. This also has circular and longitudinal fibers, just like our muscularis mucosae did. Its role is going to be more to propel food forward. So that inner layer is more about churning. These stronger, thicker, more outer layers are going to basically be helping us with peristalsis, which is kind of the movement of food through the tract as a whole. So the circular layers can change the diameter of the tract, and the outer longitudinal layer can change the length. And by working together, they're going to basically kind of squeeze alternating segments of the tract, propelling food forward. And we'll talk about that when we talk about control of the GI system as a whole. So that'll come back. It's just a little tidbit about what is actually doing the squeezing. Within the muscularis externa, we have our second part of the enteric nervous system called our myenteric plexus, or Auerbach's plexus. Again, these terms are equivalent. To help you remember myenteric plexus, it is also named for where it is, a little sneakier. So this my tells you muscular, right? Like our myofibrils, right? Whenever we see this my, we're kind of thinking muscles. And then enteric tells you just as part of the enteric nervous system here in the wall of the gut. So it's trying to tell you what layer it's part of. It was more helpful back when everybody knew Latin. So finally, we have our serosa. So this is our outermost kind of layer here. Our outer layer, basically made up of connective tissue and epithelial tissue, specialized epithelial tissue called mesothelium. Mesothelium, that epithelial tissue, is continuous with your mesenteries, which you saw in lab, are those kind of sort of fan-shaped, kind of fatty connections that were holding your GI tract to the posterior wall of the abdomen. So they're also continuous with the peritoneum, which was our kind of serous lining of the gut. And next, we're going to move through the GI tract in the order that food, like a bolus of food, would move through it, starting with the mouth. So the mouth does start the digestive process, first with mastication, which is just chewing. So physically breaking up the food with your teeth is actually very important, right? Because our enzymes are going to have more trouble getting to all the inner parts of that food if you just swallow a giant ball of it whole, right? This basically makes more surface area, makes it easier to digest. Your saliva is also really important. So this starts the sort of chemical digestion process. So your saliva has two main roles. One is to act as a lubricant, right? If you eat like a Dorito, right? If you didn't lubricate that, that would hurt going down your esophagus, right? You don't want to scratch up the walls of your GI tract. So your saliva, working in concert with the mucus that you're producing in the walls of your GI tract, is helping you swallow these things without damaging the GI tract. And then the saliva also includes salivary amylase. So remember from way back when we see ACE, that should tell us that we're thinking about an enzyme. And amylase specifically digests some carbohydrates. So it's going to be digesting starch. If we're talking about like plant carbohydrates, those are mainly in the form of starch. And it's going to digest glycogen, which we know we have in our bodies. It's more of an animal way of storing subunits of glucose. So carbohydrate digestion in particular is starting in the mouth. And I don't know if you guys have all tried this, but it was a big fad when I was in second grade to hold a cracker under your tongue and wait till it started to taste sweet. If you have ever tried this and you noticed it worked, that's what's happening here partially, is that you're starting to digest basically that starch until it actually already begins to turn into smaller carbohydrates in your mouth. At least that's my theory of what was going on. So after we pass through the mouth, we go into the throat, aka our pharynx. So this is that common passageway for food coming from our mouth. We remember from the respiratory system, we also have air coming here through the mouth as well as through the nose. That's our pharynx. Then we want to make sure that the food, instead of passing through the larynx and into the respiratory system, we want to make sure it passes posteriorly into the esophagus. So this is part of why when we swallow, we have our epiglottis kind of cover over our larynx so that the food doesn't end up in our lungs. So once we're in the esophagus, we're going to travel through a couple different regions. So a lot of places we're going to see multiple sphincters like we saw when we were looking at the bladder in our last unit. So we're going to have an upper and lower esophageal sphincter. When we see two sphincters, we're often going to expect to see that one's made of smooth muscle and under unconscious control, and one's made of skeletal muscle and is under conscious control. So the upper esophageal sphincter is the one you can control, the one at the top, right, at the base of your throat, basically. It's made of skeletal muscle. So it's controlling that entrance from your pharynx into your esophagus as you swallow. Then we have a muscular tube. So the top part, and specifically the top third, superior third of the esophagus, is going to be made up of skeletal muscle, just like that upper esophageal sphincter is made up of skeletal muscle. So that sort of tissue type is continuing. So when you actively swallow, that's part of what you're probably feeling. Then the lower 2/3 of that esophagus are going to be made up of smooth muscle. If you think about it, right, the lower 2/3 of your esophagus would be like the part traveling behind your trachea, behind your lungs, through your thorax. That part you're not controlling, right? At least I'm trying. I don't think I can, right? You shouldn't be able to because it's made up of smooth muscle. So that smooth muscle then leads us into the lower esophageal sphincter. which is also made of smooth muscle, just like that part of the tract superior to it. So that lower esophageal sphincter, what it's doing is controlling food passing between the esophagus and the stomach. We want that ideally, right, to be a one-way kind of valve, like food pass into the stomach, but not allowing acid and stomach contacts to come back up, right? If it's not working quite right, that's part of what causes gastric reflux, causes like heartburn, right? It's partially about the acid from the stomach getting back up into the esophagus, even if you're not actually vomiting, right? That's part of what is happening here. So that's what the job of that sphincter is. Next, we get to the stomach, where we're storing food that we've swallowed. Stomach can change a lot in volume, which you may or may not have seen in lab. Empty, it's about 50 milliliters. Full, it can be up to a liter. Sure, it could probably be a bit more, depending on the person. Part of the reason for this is that we have folds in the stomach that allow it to kind of accordion out. So those are our rugae. And inside the stomach, we're continuing our digestive process. Right, so we've started digesting food in the mouth through mastication, through chewing, as well as digesting carbohydrates, partially using that salivary amylase. The esophagus was really kind of just a passageway, not much digestion going there, just kind of movement. But now that we're in the stomach, we're going to really start to digest. So the stomach is going to secrete something called gastric juice, which is going to have enzymes and largely acid in it. It's going to mix together with the food that we've chewed and swallowed. When we mix food and gastric juice together, we're now going to call it chyme. That's how you pronounce that word. And so chyme is talking about this sort of liquidy mixture of our food and our enzymes and our acid, which is then going to pass through the pylorus and into the rest of the GI tract. This is also going to happen slowly or kind of at a consistent rate because the pylorus leads into another sphincter, the pyloric sphincter. So we're going to be letting little bits of the chyme into the small intestine bit by bit so that we have enough time and enough sort of space so that they all the pieces of that chyme, all the pieces of that food come into contact with our enzymes, come into contact with the walls of the small intestine. So we're going to think a little bit more about that gastric juice. So the gastric juice is actually coming from these gastric pits. All right, so this would be like the wall of the stomach surrounding our food. So this is my stomach. Okay. And the pits descend further into kind of the layers here of the stomach, where we're going to produce our acid and we're going to produce one of our enzymes. Okay. So at the top of the pit, I'm going to kind of go from the bottom of this list to the top because I think it's easier that way. So as you descend into the pit, first you're going to see these neck cells that are secreting mucus. And this is because the environment of the stomach is highly acidic. So you basically don't want to start digesting the walls of your own stomach. So the mucus is kind of going to act as a protective layer for the tissue of your stomach itself. As we descend further into the gastric pit, we're going to see kind of in the bottom here, we're going to see two types of cells. We're going to see chief cells. Those are the ones here in yellow, and we're going to see parietal cells. Those are the ones in purple. Okay. And they are also going to be secreting things. There's also a separate sort of G cell, which would also be down there in the pit, but it's not pictured here as far as I can tell. So we'll think about those. Let's do them in this order. Okay, we'll think about the chief cells first. So the chief cells, these yellow ones. So their job is to secrete pepsinogen. Okay. And whenever we see that ending inogen, like when we thought about our clotting factors, that should tell us that this is kind of an inactive form of some other type of molecule. So pepsinogen is our inactive form of pepsin. And whenever we see that beginning pep, it's like peptide, right? But this is involved with digesting our proteins. So you should associate chief cells with protein digestion. The other two things in this list are made by the parietal cells. So those are the other ones, the purple ones. So our parietal cells are where the acidity is coming from. So the parietal cells are producing hydrogen ions, which is maintaining and producing that acidic environment of the stomach, which is actually necessary basically to activate some of our enzymes. And then the parietal cells are also secreting something called intrinsic factor, which is just something we use to absorb vitamin B12. So that's a really kind of specialized enzyme, I suppose we can call it. So final kind of grab bag thing not pictured is that gastrin, one of our hormones, is secreted from the G cells. So this is one of the things that's kind of controlling the activity of our digestive system, especially like hunger cues, and we'll talk about that when we get to a sort of overall control of this system. So we'll leave off here, and I think that just means next time we'll begin with our, we have a little bit more about the stomach, but then we'll go mostly into our small intestine. All right, and the next time I see you will be Wednesday next week, I believe. I did not know we have Monday off. Very excited about it. Hope everyone has a good Easter break.